Hey everybody, welcome back again to another Journal Club brought to you by Lifespan.io. I'm your host, Dr. Oliver Medvedik, and we have yet another journal article that, of course, is apropos to the topic of aging and the reversal of aging. It is a paper from the Serrano lab and others. I believe Serrano is now at Altos Labs, if I'm not mistaken. I think I spotted that in the section of where everybody's from. Let me share my screen here for a moment. So the paper is a Nature Metabolism paper. I used to work on genetic factors that regulate silencing in genes in the genome using yeast as a model organism eons ago, but I never really worked directly on metabolism. This is a Nature Metabolism paper, and it's a topic, it covers a topic that, everybody's been talking about since 2006, 2007, which is cellular reprogramming and the OSKM factors, transcriptional factors that regulate cellular plasticity ever since they've been isolated and identified at the Yamanaka lab and have been utilized not only to generate induced pluripotent stem cells, but also to essentially turn back the clock on cells in a technique called partial reprogramming. So full reprogramming, you got cells that basically become pluripotent stem cells, and you can get all sorts of cell types from all the three different the major tissues, right? Endoderm, ectoderm, mesoderm. But if you do this for a shorter period of time, you could reprogram, quote unquote, the cells to a earlier age, and that's indicated by changes in the epigenome, most notably the methylation status, which is nowadays used as a internal clock of aging, and these cells basically look and behave from that perspective and other perspectives as younger cells. And this paper basically looks into cellular reprogramming and how it can be optimized. And they identify that vitamin B12, also known as, I believe the pronunciation is cobalamin, is a limiting factor for cellular reprogramming. So evidently vitamin B12 is necessary for efficient cellular programming, and we'll see in a few moments why. So this is the Kovatcheva et al. Paper. And here are all the institutes that everybody's familiar with. Karolinska Institute in Sweden, Barcelona Institute of Science and Technology, and I mentioned back here Altos Labs, right, which the last author is affiliated with, Manuel Serrano. So that's my brief summary. Anybody have any questions? Welcome to our group, Ali and Micah. I know this particularly interests you guys because we've been talking a lot about cellular reprogramming, particularly in mice, and this is a mouse paper.
I do like mice.
Just for a change, right? Mice.
You just say you like mice?
Yes, keep them
We all like.
alive as long as possible.
I like mice. I actually caught a mouse in my apartment last week.
Warning, tangent. Whoop, whoop, whoop.
What? Huh?
Tangent, warning.
Tangent warning? It's a mouse.
Oliver's off on a tangent.
It got caught in one of these plastic mouse traps. It caught it by its tail, so it didn't die. I took it outside and I let it loose in a city park so it can go harass another apartment dweller.
Now you're gonna have 1,000 mice. See, I have rats outside of my property, and they destroy everything. And they're the size of small dogs. I live in the tropics.
Yeah, well, we have rats in New York City too, but this was a little itty bitty house mouse, so little tiny fella.
Okay, so did you give it some B12 before you let it go?
No, I didn't. I'm hoping you found some. They eat books, and I have a lot of books, so I can't have a mouse in the apartment. Okay, end of tangent.
Normal service will be resumed.
Yeah,
We can edit that out in post, right?
that's right. I want laser beams in post. Okay, there we go.
We don't have any post. We don't edit these. These are unabridged.
That's right. Everything is presented raw and unfiltered here,
Exactly.
the way we like it. Okay, so scroll. There's a lot of supplemental data, which I won't be going into. We'll be looking at the figures. So, interestingly, they do a couple of things in with these mice, and I'm not really too familiar with like some of the connections. So one of the connections is they looked at the influence of the gut microbiome on mice. So before I do that, I just want to skip into maybe I should skip to figure 2 because that's where they actually have a summary of vitamin B12 and actually what it does, right? Maybe that should have been first. So vitamin B12 is a cofactor. So vitamins, vital amines, yes, some of them are, were identified as basically, parts of proteins. So, proteins are obviously composed of amino acids, but they have other additional factors, if you will, molecules that bind to them and basically enable them to do their job, right? Fold properly, engage in, reactions where electrons are transferred, so on and so forth. So vitamin B12 is necessary for the functioning of some of these enzymes. Two of these enzymes that use vitamin B12, or I think the only two in mammals, are methionine synthase or synthetase, which is this green blob here, which they kind of sketch with a B12 attached to it. And I believe another one, it's a methyltransferase known as MUT or MUT, which is, what does that stand for exactly? Where's the methylmalonyl coenzyme A mutase or MUT. So these are used, or particularly MS is used as basically in one carbon metabolism. So this is, cycles to synthesize amino acids or recycle amino acids. And one of them is, the main one here is methionine. So, methionine is obviously a, essential amino acid, part of the 20 amino acids coded in our genome. The first amino acid of all proteins. But it's also used as a kind of a substrate to pass along, or I guess to pass methyl groups onto other reactants such as S-adenosyl methionine. This thing abbreviated SAM, which is then used as a substrate by methyltransferases, and these are what basically these whole category of methyltransferases, obviously do methylation reactions. So methyls are basically one carbon functional groups, so a carbon and 3 hydrogens. And so when we talk about the epigenome, a lot of times we're talking about the methylation status. So, where you have CPG islands, you have methyl groups attached, or particular histones have methyl groups attached, and that affects basically the accessibility of DNA to transcriptional factors. So I believe if you have more methylation, you have more silencing; less methylation, you have desilencing. So vitamin B12 is necessary for basically generating through SAM, S-adenosyl methionine, all these methyl groups that are required to methylate things. So both the genome, as far as histones and nucleotides for epigenetic modifications, but also there's a bunch of other methylation reactions that happen on other proteins as well. Yeah, Micah.
Is B12 only required for methylation, not for demethylation, as far as we know?
Yeah, I believe so. Because a demethylase would just take the methyl group off. To be honest with you, I don't know the exact answer to that question because, I don't know if this functions as a circuit. So if you demethylate something, it has to basically then reattach to something, and then you need B12 to basically regenerate that pool. So I'm not 100% sure
Sure.
if it's required for both that yin and yang aspect, so to speak,
Right.
of methylation, demethylation, to
And
keep it efficiently going.
in partial reprogramming,
So directly no, but indirectly maybe.
right? In partial reprogramming, am I correct in my understanding that the amount of methylation that happens versus demethylation is, approximately the same-ish? Like it's not like the partial reprogramming mostly is demethylation or mostly methylation.
Um,
Like movement, basically.
yeah, well, there are changes. I think somebody can correct me if I'm wrong. I probably am wrong, but I know that there is a general trend towards. I always screw up the order of this, but if you have when you look at cells and they're aging, um, you basically have more hypomethylation, so demethylation, and I think if you have cellular reprogramming, it shifts in the other direction. And of course, maybe your results will vary at specific loci, but I think that's the general trend.
And is that true all the
I
way
think,
back? Go ahead,
sorry
Elliot.
to
Go ahead.
interrupt, but
Yeah.
like in iPSC cells, you barely have any methylation. So yeah, I don't think so. I think hypomethylation would be much more common in iPS cells.
Well,
So you have
one could imagine that there's like an optimal middle that's maximally methylated at optimal age, like 25 years old, right? And so you're maximally unmethylated as a newborn and just before you die. Could be theoretically. I don't know if that's the case.
so I asked this question specifically. Well, not this exact question, but the one question I asked Steve Horvath, who's one of the leading experts in methylation clocks, obviously, who's has the eponymous Horvath clock named after him, and others, whether he thought these changes there was any kind of pattern to them or they were stochastic, and he at the time I asked him, which was a few months ago, he thought the data pointed that at least what he was looking at, that the changes were as an organism ages, the methylation changes are stochastic in nature. So it's not so part of that is that it's not a kind of a programmed event. It's the methylation changes kind of randomly go into random kind of disarray as an organism ages.
Can I also interrupt and say something? It is stochastic, but it doesn't mean that we don't observe the same methylation changes in different organisms, different cell types over and over again.
Right.
So it is stochastic, but still, like in most genes or in most PPGs, what we observe is really like we usually observe the same things in various cell types, various people. So that's how methylation clocks work. And also, you can also, like, I think this can also have some biological relevance to it because, of course, these sites, like certain sites, certain CPGs, have something specific so that the stochastic methylation, like methylation or demethylation, constantly occurs in the same way. So probably there is at least some biological relevance to it. And there is a great article about this. I will send it to chat, and if you are interested,
please do.
Please, you can check it out.
All right. So yeah, stochasticity, randomness with constraints. We generally lose hair as we get older, but it tends to be patterned in a certain way as the hair is lost. Okay. So one thing that they looked at before they kind of switched to the kind of need or the limiting role of B12 in maintaining or optimizing OSK reprogramming is they implicated also the gut microbiome. Now, I don't know if anybody else read the paper. I'm a little vague as to the major role of the gut microbiome to providing vitamin B12 because our cells can't synthesize vitamin B12. They need to take it in from the environment, but your microbes in your gut, parts of your colon do synthesize vitamin B12. But from what little I read, that's not actually accessible to the organism. It's accessible to the bacteria. So if they're cranking out B12, it's not really accessible to the to the mouse, let's say, or the human. But that being said, they've done experiments here. So this is figure 1A, where they basically implicated the necessity for a healthy microbiome for proper cellular reprogramming. And what they did was, and again, maybe somebody could shed light as to how this exactly connects to the rest of the experiments here. But anyway, I'm just going to present this because the data's here. So they treated these mice with ABX, which is basically a cocktail of several antibiotics, I think vancomycin and a couple of others. And this basically, I would say probably wreaked havoc with the gut microbiome. And then at day 0, where they started, so these mice were transgenic mice, I believe, where OSKM was integrated at some promoter that was doxycycline inducible. So that's another antibiotic, which is, or a derivative thereof, which basically allows this promoter to switch on and you get OSKM expression. And you can basically have it on, for as long as you're maintaining doxycycline in the water or the chow of the mice. And while they did that, they either supplemented or did not supplement with vitamin B12, but they also maintained ABX. So basically, wreaked havoc with the gut microbiome, and then they analyzed the mice after euthanasia at day 7. So what they noted was, so here's a bunch of histological sections. Let's see, this is colon. So they looked at colon, they looked at pancreas. They also noted that in mice, at least, the reprogramming, or at least, the level of reprogramming, even though doxycycline, I believe, is accessible to most, if not all, organs. And they actually, in the paper they mentioned, they looked at expression levels of OSKM, and that was fine in a lot of the organs, or most of the organs they looked at. But the degree of reprogramming was different. So there's some organs are more, more easily reprogrammed than others. Some tissue types, I believe the colon was more easily reprogrammable, and the pancreas. They mentioned the kidneys were particularly recalcitrant to reprogramming. And I don't know off the top of my head why that's the case. Obviously has something to do with accessibility of the particular regions of the genome that probably need to be reprogrammed, if anybody has any input on that, but there is a difference.
Which of these colored bars is the doxycycline without ABX, and which ones with ABX? Like which ones with a healthy gut microbiome, which ones are without?
Are you asking me about the figure, or are you asking me about the question I asked?
The figure on the right.
Oh,
in the blue one maybe is.
Oh, I didn't.
No.
Yeah, I didn't get into that yet. Are you talking about? So the figures on the right is basically a summary of what you're seeing on the left.
Right.
So there's the bar, the graph, which basically tells you what's what.
I see. Okay.
So you have wild type, you have OSKM, minus, no so nothing, no ABX on this side, then plus ABX over here, doxycycline. So what do they do here?
Figure is better in the charts.
Yeah. So hematoxylin, eosin staining, so general staining of proteins and structure of the cells in the colon. And then they use 2 different markers of partial reprogramming, which is something we might look into for our project, Micah Ali, which is SCA1 and KRT14. So these were, one of them is kind of an early marker, one's an intermediate marker for reprogramming. I think SCA1 might be a transcription factor. I don't know how tissue specific it is. So wild type obviously is just wild type, so controlled doxy, that shouldn't do anything. And then OSKM, when they do induction, if they add doxycycline, the staining is showing up in portions of this, areas of the cells in the colon. Let's see, they do the same thing, get to the pancreas. But that seems to be severely curtailed when these mice are treated with this, with this cocktail of antibiotic. And this is basically just, percentage tissue that they note. They do note, though, that, let's see, so OSKM, doxycycline, ABX. So you basically get a, a blocking, if you will, I guess, of cells that are indicating, partial reprogramming is taking place. And I'm not 100% sure of the significance. It seems that, you do need a healthy microbiome, but I'm not really 100% sure how vitamin B12 ties into it, because initially I thought, well, you ablate it and you lower probably B12 levels that are available from the microbes in the gut. But they do mention later in the paper that when they looked at the shift of the microbiome after doing this, it shifts to a less heterogeneous cell population of microbes, but they shift towards microbes that tend to produce more vitamin B12. So I'm not too sure about that, what that's supposed to imply. If anybody read that.
You're saying that after the ABX treatment and the partial reprogramming, the new gut microbiome was proliferated with bacteria that produce B12? Is that correct?
Well, I'm not saying that. I'm just reading it from the paper.
I understand what your interpretation of the paper is.
Yeah. So let me get the page here. Let's see figure. Where's figure 1 in the this observed change in the gut microbiota could be indicative of a systemic deficit of B12 affecting all microbiota, but the entire.
My suspicion for why they talk about the B12 is maybe that's just how they got here. Like they originally did the test of the ABX and found that they noticed B12 production up afterwards, and they're like, huh, that's odd. And they tried doing OSKM with B12, and like, oh, look at that.
Yeah, except it wouldn't be in the blood, I think, because it doesn't. It's just limited to the. Anyway, if the kind of reviews I read are correct, then it's not really getting in. So we found microbial changes consistent with this phenomenon. The few genera of bacteria able to synthesize B12 were generally enriched in OSKM mice after 7 days of doxycycline. Okay, after 7 days of doxycycline. Never mind. Okay, not after ABX. Sorry, I had that wrong. So so what this might be actually happening is kind of not so. All right, so if you use OSKM, you're probably depleting vitamin B12, and some of that vitamin B12 is probably required to maintain a healthy gut microbiome. So then if you do OSKM treatment, you shift towards a population of bacteria, and they mentioned Proteus, E. coli, Salmonella, to kind of reestablish vitamin B12. So this is more in line of keeping the microbiome healthy rather than the microbiome itself
I see. So
interacting with methylation happening within the genome.
So the hypothesis here is that the OSKM expression results in B12 depletion. Your microbiome now is also depleted of B12 because it's not getting as much from the body, and so the population within the gut basically shifts to fulfill its own needs.
Yes, that's my understanding. Now that being said, it seems that if you screw up the microbiome, you also tend to screw up reprogramming. But in that direction, I'm not really sure how the microbiome is helping reprogramming. Does that make sense?
Yeah, I mean,
unless I'm missing a piece there. Yeah.
Maybe they're also giving some B12 to the body as well.
Maybe, but I don't know. We can Google that here. But when I looked it up, it seemed you know, the reviews that I looked at basically were stating that the B12 generated in our gut microbes are contribute an insignificant amount to
It
our
could
blood
be,
serum levels of B12.
sure, it
But
could be
sorry.
it's the B12 consuming bacteria that are helpful in partial reprogramming. And when the partial reprogramming depletes B12, that population starts to die off, and now the partial reprogramming is in spores. It could be indirect like that, where it's not that you're getting B12 from the gut, it's that the gut needs B12 to do its job, and part of that job is helping the partial reprogramming do its job. And like it's a kind of a circular thing. So partial reprogramming consumes B12, the gut runs out of B12, and so that population of B12 consumers dies. That population of B12 consumers that dies now is unable to provide the system with whatever it was they were providing that helps this partial reprogramming. I don't know what that other thing is.
Yeah, that makes sense.
I think that makes sense. Also, in the case of a B12 scarcity in the body, maybe in that case, somehow the body starts getting B12 from the microbiome, because like in scarcity, a little amount of B12 can change many things, or maybe that would be the one explanation, but I don't know.
Yeah, that's reasonable. It's like under normal conditions, your body doesn't get B12 from your gut because it gets it from everywhere else. So, which is why the papers would all say, the gut B12 doesn't go through your body. I guess the way to answer that would be to find out if there's anyone who's done any research on B12 deficiency and B12 in the gut. Like, can you have high gut B12 while being B12 deficient elsewhere? Or is that not a possible state to get into? If that's an impossible state to get into, then that suggests that the body can pull from the gut, B12 from the gut, if it needs to, but normally it doesn't. Because I think B12 is one of those vitamins that it sits in your fat tissue for like a decade or something. Like you just need a little bit of B12 once a decade, and you're generally fine for your normal needs. And so my guess is that people, I don't know, how common is B12 deficiency? Is this something that happens very often? My understanding is it's pretty rare, right?
One of the B vitamins, I'm not sure if it's B12. I know like there's some conditions, especially people who are alcoholics. There's a deficiency in one of the B vitamins. It might be B12, could be B6. I'm not sure.
So I think later on, though, they do actually talk about B12 supplementation and OSKM. Is that right?
Yeah, actually right here.
Okay.
So anyway, this figure C is basically they do, I think, a genome-wide analysis in the microbiome to see what the shift is for particular genes, and I think the predominant ones were, shifted towards cobalamin metabolic processes. So basically, shifting towards vitamin B12 synthesis. So that's kind of, I think, how vitamin B12 got partly on their radar for the gut microbiome. Here's additional evidence showing, so they look at serum B12 levels. So they do go, noticeably significantly down when you doxycycline treat OSKM transgenic mice. So, you have expression of OSKM. So there's a great need for methyl groups. So the B12 levels actually noticeably drop.
My reading of charts is very bad. They're saying there's no statistical significance between the untreated and the wild type doxy. Is that correct?
Yeah, they're saying there's no statistical difference. Yes.
Okay.
Yes, you see a trend. Maybe doxycycline is doing something, but according to this NS, if they look at the numbers, there is none. Maybe if they had more mice, there would be a slight decrease. I don't know off the top of my head what doxycycline itself would do to the gut microbiome.
It's an antibacterial, right? I would assume that it would wipe it out.
Yes.
Just like AGX.
Not as potent as that ABX stuff, which is like 4 or 5 antibiotics. And I don't know if the dosage is high enough to actually, really mess up the microbiome. Anyway, so they look at also the pancreas, and these two figures here, OSKM and then OSCAM plus B12 supplementation. And what they note here is that they look at these factors, SCA1, KRT14, which are indicative of intermediate reprogramming, and NANOG. And in the time frame that they looked at, I think this is day 7, I'm not sure. You have, using at least this histologic technique to measure for these factors, there's a significant increase in factors that are indicative of reprogramming taking place when you supplement with B12 and you OSCAM treat these mice. So
did they test the mice to see what their B12 levels were like before this? Like, did these mice, are they confirmed to have whatever healthy levels are for a mouse? I'm curious if this is a situation where they brought it up to healthy or they exceeded normal healthy values with the supplementation.
Yeah, good question. I don't know. Somewhere, the question reminds me of when people do lifespan analyses of mice, right? Where you do some sort of change to a mouse and you get it to live longer, and then the question is, well, was this mouse short-lived to begin with, and you just kind of repaired something and brought the mouse back to normal longevity, or did you take a normally lived mouse and actually truly extend its average and maximal longevity past what it normally is?
Same question.
So yeah, I don't know. It would meet in the materials and methods. I would, I don't know how hard it is, how lengthy it is to continuously, or how it, to measure serum B12 levels. I'm assuming it's fairly straightforward, but I don't know if they measured the baseline of B12 for all of these mice that they had.
I also wonder if, I don't know anything about mice biology, but in humans, B12, like I said, sits around, I believe, in your fat tissue for a very, very long time. And so you can take like a bolus dose and you're good for a decade. I'm curious if mice are similar, and if so, could you give them, a bolus dose of B12, wait 6 months, and then do partial reprogramming, and they do just as well as if you gave them the dose later on, because they had all that stored up. Like, is the body able to recruit that B12 out of storage for this process, or does it need it in circulation at the time that the OSKM is delivered? Like, does it need to happen simultaneously?
I have no idea. I think we need a mouse nutritionist right here to kind of join us, who can basically answer those questions. That's something I don't know off the top of my head. What how much latent stores of vitamin B12 a mouse has in its fatty tissue, and how much saturation we can have of vitamin B12. No clue, unfortunately. Does anybody know the answer to that question out there?
If not, some research next team can do when they're researching B12 and OSKM.
Yeah. These are really fundamental questions when it comes to mouse, and also human, like nutritional metabolism. I'd be surprised if there aren't a whole bunch of, again, early papers where they basically, because that's something that fascinates a lot of people, which is, how we get proper nutrition, nutritional intake, and to get all of our systems operating, blood plasma levels of various vitamins and other metabolites, and how long they basically circulate, the half-lives and so on and so forth, where they're stored. I'd be surprised if there aren't fundamental physiology papers out there that basically just, at least for some strains of mice, really nail, how long and how much vitamin B12 stores there are in particular strains, and how that is reflected in the fat content of a particular mouse, and so on and so forth. It's probably a lot of nutrition papers out there on this.
I'm sure. They do have one quote in here that says, however, we did observe a significant increase of vitamin B12 stores within the kidney after supplementation, indicating that B12 absorption, distribution, and storage were occurring normally. Suggesting that, this could all just be a dosing issue, but suggesting that either they dosed more than was needed for the partial reprogramming to occur, and so the leftovers then went into storage, or it suggests that Well, it also may suggest that they were in fact deficient. Like if there was enough, if the body still was storing more B12, it kind of suggests that it didn't feel it had enough. Like, 'cause I believe that while your fat will store B12 for a long time, it has an upper limit, and then it stops storing it at a
Yeah,
certain point. Like
yeah,
regulating.
and I don't know if that's beyond normal or not, because I'm pretty sure I could store a lot more fat in my body than I have right now, but I'm not going to attempt that. And
Sure.
I
Yeah.
don't, so I don't think, despite the fact that, my girlfriend says I'm 20 pounds overweight, but, as I think maybe I'll use it as an argument, say, actually, I'm at an abnormally low level, and I can safely pack in another 100 pounds of fat.
That's fair. That's fair. Just because your body can store more of a thing doesn't mean that that's necessarily the ideal levels.
Yeah, so I don't know. I mean, you might be right about that. And I'm talking about apples and oranges, kind of. This probably doesn't translate from vitamin
And
B12 storage to
it's a
fat per
good
se.
it's a good case study at the very least of how that argument does not hold up.
Okay. Right. So what do they look at? So we briefly dipped into figure 2 to look at this diagram here. So again, this is tying into vitamin B12. So tissues undergoing in vivo reprogramming exhibit an increased demand of 1C metabolism, 1 carbon metabolism. So this is basically related to the moving around of single carbons, particularly as methyl groups. So they showed the folate methionine cycles that we quickly looked at there. And let's see, B, changes in metabolic pathways. So they looked at, so this is pathway impact. Don't really know exactly how to analyze it. Well, all of these circles are supposed to represent different types of, I guess, clusters of genes that are regulated, that are related to different processes. And the most changes that happen, is this during OSKM, reprogramming, are in this glycine serine threonine metabolism. I guess some of the others probably are associated with that, but anyway, this is basically these pathways here are also tie into the single carbon pathways. So when they were doing this, they basically noted that these pathways that are involved in single carbon metabolism go up with OSKM, probably to help replenish the levels of methyl groups in the form of SAM, which is S-adenosine methionine, which is basically how the methyl group is passed on from one enzyme to another. So this is basically, what did they look at? I think this is a serum test. So let's see, blood serum, I think. C, full change of SAM, so S-adenosine methionine to methionine ratio. From B, what's B? I think this is from serum. So they do a mass spectrometry reading to show that, so the ratio of SAM over methionine basically goes up, since when you have doxycycline treatment. So basically everything is basically being shunted over to the methyl groups into SAM. So OSKM can basically use this for presumably that's what's happening for more reprogramming. And they also look at some of the expression levels of some of these factors. So this is I believe a Western blot, looking at the pancreas. So this is induction. So is this days induction? So MS is methyl. So this is the methionine synthase. So let's see. Pancreas, what are the 1, 2, 3, 4? Vinculin is a loading control. Is this day 0, 1, 2, 3, 4? Let's see what those different wells are. Immunoblot, NRTQ, PCR, and pancreas. I think this might be just 4 different samples, 4 different mice probably. I think there's 4 different mice in day 7. So noting that you basically have also an upregulation of the enzymes that are to kind of recapitulate. So basically to produce more, to keep this pathway flowing, to produce more more accessible methyl groups for OSKM. They don't notice So I guess there's already a high level in kidney, and kidney was the one that they mentioned was recalcitrant to reprogramming. So yeah, you would notice a change, from low to high in the pancreas, which is basically undergoing reprogramming. But looks like it's already high in the kidney, and it doesn't change, at least the methionine synthase enzyme. So I don't know if this basically means that why kidneys are particularly recalcitrant to reprogramming or hard to reprogram. It appears you have OSKM expression in kidneys, you have high levels of methionine synthase, you probably have enough methionine or enough methyl groups available for reprogramming to happen, but so it doesn't appear to be for lack of substrates and factors for reprogramming to happen in the kidney. And I don't think they really talk about that in this paper, why there's a certain tissue specificity to difficulty in reprogramming, particularly in the example they gave with kidney. If anybody has any comments on that.
I do have a comment on this figure before we move on, though not that specifically. Figure F there shows it looks like they did the B12 as a bolus dose after they started the OSKM, if I understand that correctly.
Yeah.
Which suggests to me that I would not expect the microbiome bacterial colonies to change significantly over the course of one day. And so the fact that they gave the bolus dose like after they started reprogramming and like a day later, they sacrificed the mice, I believe, if I'm reading that correctly. Or rather, very late in the game, basically they added the B12. That suggests that it's not that the B12 is changing the bacterial population. There's not enough time to change the bacterial population. Like, you're not going to get all the B12 consumers to all of a sudden show back up and repopulate in 24 hours, which kind of writes off my theory before that the B12 was feeding the B12 consumers in the gut, and then those were then doing something useful.
Yeah, I mean, I don't know how fast the shift could happen, because certainly, at least I don't know how optimal the gut is. I mean, certainly under optimal conditions with, in a tissue, in an incubator, E. coli at least, replicate once every 45 minutes to 50 minutes. So 24 hours, that's a hell of a lot of generations.
That's fair. And you think that's enough to potentially change the population dynamics within a group colony?
Yeah, but I think what they did with the bolus here was to show that they let's see, what did they do? Because again, when they're giving B12 to these mice, I think it's through either water or food. So they wanted to show that the absorption of the gut somehow wasn't affected by what they were doing. So let's see, figure F, they talk about it here. Okay, yeah, here it is. Finally, to ensure that the depletion of serum B12 levels and its associated low levels of methionine were not simply caused by defective oral uptake due to the reprogramming of several digestive organs, because the colon gets reprogrammed pretty quickly, we administered a large bolus of vitamin B12, here they mentioned 100 times the recommended dietary allowance. On day 6, before euthanasia, mice expressing OSKM had significantly higher levels of B12 than wild-type mice following the bolus, which is known to occur in B12 deficient rodents. Further indicating that reprogramming does not compromise oral bioavailability. Strikingly, the bolus rescued the depletion of MET levels in the serum.
I see. Okay, so this wasn't this didn't show improved partial reprogramming in this particular test.
Yeah, they basically wanted to show that that because you're getting reprogramming everywhere, and supposedly the colon kind of gets reprogrammed more easily, that this wasn't affecting vitamin B12 absorption, at least according to this bolus test. I don't know how, this is 100 times the dose. So yeah, right. I mean,
so my hypothesis is back on the table then.
Okay. Let's see. Figure 3, vitamin B12 supplementation enhances. So So histone 3, lysine 36, 3 methyl group. So basically, this is one marker of reprogramming. Is basically the I think the trimethylation of this particular histone. So it enhances the methylation of this, and has a cell autonomous role in reprogramming. What does that mean? So they're looking at MEFs, murine embryonic fibroblasts. So this is in vitro now. So what we were looking at before is in vivo in mice, and they're now looking at reprogramming using fibroblasts in vitro, and showing that you can basically spike B12 in and basically improve reprogramming. And this assay that they use are basically, you get these. So this is going into full reprogramming into iPSC cells. So you get these kind of undifferentiated like foci of cells that cluster. So these iPS colonies, and they showed essentially if you add in B12, it improves the efficiency of OSKM, at least the murine embryonic fibroblasts. Yeah.
These all have OSKM? I mean, that chart shows only the first one has OSKM, the next one's just B12.
No, the OSKM is down here, I believe.
Oh, I see. Okay, so every single bar on this chart has OSKM. The first one has only OSKM.
Only OSKM, yeah. And then they supplement B12, then they supplement with an inhibitor of I believe an enzyme that basically helps to resynthesize MAT2. So basically it's an inhibitor that basically blocking the synthesis of in, I think somewhere in 1-carbon metabolism. So you basically what's MAT2A specifically? We should just spell these out.
Yes.
Anyway, it's an enzyme that basically can replenish either directly or indirectly the levels of accessible methyl groups. So you're basically blocking the ability of B12 to work.
It's methionine.
Yeah.
Adenosyltransferase 2A inhibitor.
Okay. Yeah. NSC. It's another. I think it's a demethylase inhibitor, and I'm not sure if this is supposed to act as a negative control. Interestingly, which probably shouldn't surprise us, but directly adding in, and this is available as also a supplement, S-adenosine methionine, which is basically the main substrate for methyl group transfers. That really helps iPSC colony formation. So really accelerates, enhances the ability of OSKM. That being said, I don't know if anybody else has looked at just supplementing SAM or SAM into the media of mice to see if it improves the OSKM ability to reprogram it, particularly in recalcitrant tissues. Does anybody know?
I was going to ask the same thing. I wonder if B12, being a vitamin, it's easy to just ingest it and it goes everywhere.
Should be the same with SAM too.
You think so?
Yeah, I mean, you can get. I don't know what the oral bioavailability is, but you can certainly get I'm pretty sure you can get S-adenosine methionine supplements.
Yeah, I'd be very interested to see if that helps.
Because because B12 is it works on this enzyme MS and the other one mute, but SAM is basically right here with methyltransferases that you're basically altering vitamin B12 levels to get to this, to increase this concentration, at least for cellular reprogramming. This is fundamentally what you want to go up, and B12 is just indirectly allowing you to do this. So yeah, that's at least a twofold. Looks like roughly a twofold increase, right? So something to kind of look up while we're looking at other reprogramming papers to see if various supplements, particularly S-adenosine methionine basically can, improve the functioning of OSCAM, particularly to get into to reprogram more difficult to reprogram tissues. I don't know. Anybody out there look up anything along those lines?
I'm checking quickly right now, but nothing's jumping out as someone having tried this.
Yeah, so Sven mentioned a little earlier, there's a new paper that shows that in vivo reprogramming causes death through hepatic and intestinal failure. Yeah, probably not so surprising. I think they mentioned colon is pretty easy to reprogram, and I think maybe hepatocytes too. So yeah, if you get anything long enough, right? If you get teratoma formation and your cells basically lose their differentiation, then the first ones to go is basically where you're gonna get the failure. Those will be your first failure points. Just going through the chat here. Interesting question from Ali. Also not related at all, but considering the importance of B12 in memory, and I don't know anything about B12 in memory, by the way, whether it's direct or indirect, right? I mean, obviously, if you're starved in general, it's going to affect a lot of things, including memory. Could this be the reason why? Do we store our memories in our epigenomes? I don't know. There's some intriguing, maybe, right? I mean, there's some drugs out there that supposedly can reverse or block PTSD, and somebody somewhere commented, I don't know who it was, that that almost seems very suspiciously like cellular reprogramming is taking place, right? Because if you take, it's almost as if you sort of like reset something, right? And you think at it from a reprogramming aspect. If you can basically, well, it doesn't have to be at the genome level, right? So you could certainly strip a ligand off of a receptor. But yeah, I don't know, Ali. That's an interesting question. Whether or not B12 has anything to do with memory for consolidation and reprogramming, or has anything to do with that.
I also don't know. I just thought it's something interesting to think about. And also, if that's the case, it would mean that we cannot reprogram our brains to make them younger. I don't know if it's true or not, but probably we wouldn't want to lose our memories.
That's something that could be tested.
Of course.
I mean, it's a good question, and that's something that could be tested with mice too, right? I mean, do OSKM mice lose their memories if you put them in a maze? You should be able to set up some memory tests to see whether or not reprogramming has an effect on previously learned abilities.
There's been surprisingly little research done that I've seen, at least, on reprogramming of the brain. I know at least one group that is trying to find brain-specific reprogramming stuff, but I haven't actually seen hardly anything published about it. My understanding is that the brain doesn't reprogram quickly, so if you do systemic reprogramming, generally the organism dies before you get to the brain sees any noticeable changes. I don't know, has anyone seen any papers where they've tried to just target the brain somehow?
I may have come across some recent papers where they looked at promoters for OSKM that are more efficient in neurons. I'll see if I can try to get the specific ones. But I don't know how far along they went with that in vivo in mice.
Gotcha. I think that would be my guess is the reason no one's tried what you just described is because no one's been able to reprogram a brain without killing the mouse first due to liver failure or kidney failure or hepatic and intestinal failure, as Sven said.
Exactly.
Right. Unless you just have OSKM expressed only in neurons and in neuron specific manner, and basically just focus on that.
Right. which would require, I think, a new mouse line. And I think everybody right now is using the same mouse line that's got the docs-inducible systemic OSKM.
Yes, you can just create a gene network using a neuron-specific promoter and a docs-inducible system like that. But yeah, I don't think anyone's ever done that yet.
Okay, let me stop sharing here for a moment. Yeah, I just quickly wanted to see if I had this paper because I looked at. Let's see. Let's see, where's this? I'll pull it up later. I thought I had the paper that I was basically. Oh, what's this? Efficient generation. Oh, this is 25 with consistent transfer feast. Okay, never mind. Back to the paper.
It's a day of tangents.
Well, I mean, it's okay. Tangents are good, especially if that's what we do at journal clubs, rather than just plow through, right? We want to engage in educated discussion. Okay, so in vitro, it seems to be working. What do they look at here? Total SAM pool. So this is an assay that they used for, so this is a carbon-13 based labeling assay to show that, I think this is intracellular, that when you supplement with B12, you basically boost your levels of S-adenosine methionine using this assay. Yeah, fraction of total intracellular SAM13 labeled. So basically if you add B12, so not only is it like happen in vivo, but this is a cellular intrinsic process of B12 supplementation. It will, and also SAM directly, right? So in at least in murine embryonic fibroblasts, and I'm going to assume that it's probably not cell specific, that this is what's in general happening. It'd be interesting to see if the more this whole thing is also being recapitulated in the recalcitrant cells that are really hard to reprogram, because then that might shed some light as to why they're so difficult to reprogram. Because if vitamin B12 is getting into these cells and S-adenosine methionine levels go up, and you have good OSKM expression happening in these cells, but they are just still difficult to reprogram, then there's something happening downstream that's hard for OSKM and downstream factors to reprogram. And I'm not sure what that is exactly.
It seems like this SAM discovery alone, assuming this paper discovered is not already discovered, should help the iPSC generation people. Like all the people that are trying to generate iPSC is just always make sure you've got SAM in your media.
Well, I mean, just looking at this, right? OSKM here and there. I mean, that's a huge difference.
Yeah, massive.
So, yeah. So I mean, like of all the things in this paper that piqued my curiosity, the SAM one was definitely up there. I think that was the kind of the, at least from my perspective, the most kind of important thing. Maybe it was noted before somewhere, but at least in the review papers I was reading, I haven't come across any sentence that just blatantly said, yeah, make sure you have adequate SAM supplementation when you do this. Like,
which seems so simple.
Yeah, I haven't come across that sentence, to just explicitly state that. Maybe it's just so obvious that if we pull up I don't know, Ali, maybe you can tell me, is it like a standard protocol for OSK reprogramming that, every method basically says absolutely do not omit the S-adenosine methionine or vitamin B12 from your media?
I have no idea. I need to check the medium ingredients, but I have no idea. I'll check it.
Yeah, okay. Something to keep in mind. Okay, so day 3, day 5, day 10. So B12 minus, so no B12 plus with B12. I think what they were noting here is that when they were looking at the methylation status, and what were they looking at? This is like bulk methylation status, so I don't think this is a particular locus that they were looking at. The trimethylation at these histones goes up. So this is not very, with vitamin B12 supplementation. So what's the difference between, so D is, so that's total histone 3, and that's methylated histone 3. Let's see, what's the difference between C and D? Oh, they're basically showing that, yeah, you can block these effects using this inhibitor that basically lowers the accessibility of the methyl groups. And the levels go up again significantly. So not only you get more iPSC colonies, but also you have more. See, do they have this as a bar graph? I don't think so. I'm just looking at this this Western blot here, just by eye. It definitely looks like there's much more overall bulk. Histone 3 that's trimethylated when you add SAM, which is not surprising. Let's see, what's set D2? These levels go up. I keep forgetting what some of these are. Oh, that's the actual histone 3 lysine 36 trimethyltransferase. So that's the enzyme responsible for trimethylation. And these levels of set D2, which encodes that enzyme, do go up with OSKM treatment. And what do we see here? So they notice with B12, I'm not a histologist, so one thing that they mention is when you do cellular reprogramming, at least for maybe, enough to, I don't know if this is partial or getting into full reprogramming, but you have cells which are dysplastic, which basically means they look aberrant, which means like they're undergoing some morphological changes indicative of dedifferentiation. And these little speckles and dots here are supposed to represent staining for, immunohistochemical staining for lysine 3 that's been trimethylated. And these little dashes here that circle these areas are supposed to be, out of all these cells in the pancreas, those are identified visually as dysplastic. They do look a little fainter. I kind of zoomed in. I don't know if they have just larger vacuoles or what makes them dysplastic. I'm not a histologist here. They certainly look like they have some vacuoles in them that make them a little bit more different from these other cells in the pancreas. But these little like zones. But I'm going to take their word for it that those are the cells that we need to concentrate on because they are most likely reprogramming. And out of all those cells, if you add in vitamin B12 supplementation with OSKM treatment, more of those dysplastic cells are also exhibiting more of this, methylation at lysine 36 on histone 3. So indicating that, so this is pancreas now. So this is now going back in vitro. So, right, so we were in vivo. So this was all in vitro. And let's see, then I guess this is back in mice again. It's got to be back in mice again, of course. Yeah, pancreatic tissue from OSKM mice. Okay. Let's see, what do we have here? Okay. Now figure 4. Okay, I think they're looking at, so when you have OSKM treatment, there's something that they mentioned here that I don't, so I guess this is kind of an abnormal process, but they mention cryptic transcription, where you basically have, areas of the genome that are outside of the promoter basically end up, so when you have a promoter, obviously you have a messenger RNA being synthesized, right? And it starts at a particular specific area known as the +1 site. So it's your promoter basically helps line up your RNA polymerase, and any other transcription factors, and you have a transcript come off from this area. And I think when you have cellular reprogramming happen, it gets a bit sloppier, if that's a good word to use, where you have the transcripts tend to be longer than average, if I'm reading that correctly. So it's referred to as cryptic transcription, because I guess the amount of reprogramming that happens, allows for kind of not as high fidelity as binding, if I'm interpreting this correctly, of RNA polymerases and other factors. So yeah, it's a question here. But no, Sam. Oh, we're talking about the culture media. Okay. So so I'm not sure. So what I'm getting at here is the data that they were showing here when they're doing these I think they're doing RNA sequencing. They mentioned that vitamin B12 supplementation reduces cryptic transcription during in vitro programming. So they do H3K36Me3 chip, so chromatin immunoprecipitation sequencing. And I'm not really sure why exactly you would get less of these cryptic transcripts if you supplement B12 with OSKM, unless, yeah. That's something that I'm still kind of puzzling over. It seems to suggest, the data, that if you supplement with B12, it makes reprogramming, obviously more efficient, more effective, as we saw previously. You have more, availability of S-adenosine methionine, right? So that's good. So everything can progress better. And if you have OSKM, this analysis here in Figure A, so this is looking at, I guess, so chromatin immunoprecipitation addition as indicated. So they're looking at most highly expressed genes, top tercile. And it seems to be that, yeah, I'm not sure exactly how, yeah, Micah, go ahead. You have a question?
Yes, but it's unrelated to what you're talking about, so I'll let you finish your thought.
Yeah, my thought is that this entire figure here talks about how this cryptic transcription levels are suppressed when you add vitamin B12, and they're increased when you just have OSKM. And I'm not sure why the B12, if it's making OSKM more effective, why you wouldn't have the opposite result rather than have less cryptic transcription happening, right? If cryptic transcription is a consequence of reprogramming, then should more
Is cryptic transcription a good thing or a bad thing?
Well, here they're positing it as a bad thing because it's happening in areas that really supposedly aren't really necessary for the reprogramming to happen.
I wonder if maybe you think of it like OSKM, when it gets expressed, it kind of as part of the reprogramming process, perhaps is not super precise, and B12 helps it be more precise, for lack of a better analogy. Like it's helping it aim, so to speak.
Yeah, I don't know, because if those kind of rough charts are to be believed, all vitamin, really what's happening is you're just increasing the pool of available S-adenosine methionine, and if that's the case, then you basically you're just basically giving more raw material for the OS. Well, not directly to OSKM, but whatever it's expressing, because these are transcriptional factors. So downstream you get increasing methylation. So I don't know how that would basically tie into increased specificity, is what I'm getting at.
My confidence that we know everything that B12 does in a human or a mouse is very low. what I've learned over time is that every time we think we know what something does, we find out it does 10 other things later. And so
Yeah,
we know what
I mean,
B12 does in terms of that little chart they throw at the beginning, but my guess is there some other stuff that's going on as well? Because the body likes to reuse.
yeah, so I'm going to basically defer to what the authors wrote here. I don't know really if they kind of elaborate on that. Interestingly, 72 hours of OSKM induction caused a decrease in the amount of H3K36Me3 across gene bodies relative to all reads, which was prevented by vitamin B12 supplementation. H3K36Me3 plays important roles in maintaining transcriptional fidelity. Okay, one such mechanism active in mammalian stem cells is to prevent illegitimate transcription initiation outside of promoter regions at intragenic cryptic start sites, particularly within genes. We thus ask whether the amount of cryptic transcription was changed during reprogramming. CT can be estimated by calculating ratio of expression from 3' exons, defined as those from the fourth to the penultimate exon, consistent with the reduced levels of H3K36Me3. Global CT significantly increased cryptic transcription during OSKM-induced reprogramming as compared to the non-induced MEFs. Remarkably, so I guess that surprised them, remarkably, supplementation with vitamin B12 during the 72 hours of OSKM expression reduced the amount of CT to basal, that is, non-reprogramming levels. This effect was dose-dependent with respect HMF. This data suggests the high demand for methylation that occurs during reprogramming causes a deficit in H3K36Me3 that results in increased CT, which can be rescued. So I don't know if this is a hypothesis. So if I'm reading this statement correctly, we have such a depletion of the availability of methyl groups that there's some system out there that's recycling the methyl groups from other regions in the genome to be more accessible to OSKM. So basically the reprogramming can happen in other regions. I don't know if I'm interpreting that correctly, but it almost seems that that's the hypothesis, and I'm not sure if that's a known fact or whether the authors are hypothesizing that this is what's happening here. That's why you have the depletion of methylation because of H3, histone 3 at these other areas, because there's some sort of enzymatic recycling system that is basically shunting away methylation from those areas to make it more accessible for reprogramming at other areas. And that by basically supplementing with B12 and probably with SAM2, you can basically prevent that from happening. And maybe that sort of depletion is also maybe acting as a block to more efficient reprogramming. I don't know. That's my take on it. Anybody want to have a rebuttal to that?
Seems reasonable and relatively easy to test. If it is just that one pathway, then supplementing SAM, like you should have the same effect, right?
Yeah.
Right. And if supplementing SAM does not have that effect, then it means the B12 is doing something else that we don't know about.
Yeah.
Can I ask something before we move on?
Yeah.
I don't exactly know what cryptic transcription means, so
I don't exactly know
know
what it means either, other than basically it's transcription from beyond regions where it's not supposed to be transcribed.
Okay, so is it like an imprinting related event? Or like what does it mean in general? That's such a broad statement, right?
Yeah, I know. I think it means, again, we should probably have a more specific definition, but based on what the paper suggests, it's basically you have areas that are silenced through methylation, probably in this case through methylation, but probably silenced through other changes in the genome. And to put it bluntly, your RNA polymerase will fall off before it starts to transcribe those regions. But if you have a depletion of methylation or other types of silencing markers, then your RNA polymerase will basically accidentally go go into those areas further 3' while it's making a so your transcripts will be longer than average or there might be areas that are being transcribed that shouldn't be transcribed as well.
Okay, but like, I think it makes sense, but are we looking at the pre-mRNAs or mRNAs here? Like, are these mRNAs already like cut, or are these?
I think what they're looking at for A is not, in this case, not mRNAs, but they're doing chromatin immunoprecipitation of particular regions where there's promoters, and they're looking at the methylation of this histone in these chromatin IPs. Yeah.
Yeah. So we just see an overall decrease in methylation, but it could also be like the case that because B12 is also a regulator of a methyltransferase. You mentioned one methyltransferase, right?
Mm-hmm.
So like this could be totally unrelated to the reprogramming as well. So maybe that methyltransferase is responsible for methylating H3K36, and like maybe it's not related to reprogramming efficiency at all. How do they conclude that this is somehow related?
A quick search on the internet suggests that there is some data suggesting that this CT happening is potentially causal for aging, at least in yeast. If you can stop it from happening, the yeast live longer.
Yeah, that's the first time I'm hearing about this for yeast. So I have no experience with the role of cryptic transcripts. I mean, the only kind of, there were some papers that talked about, areas that are being expressed, particularly like, ancient retroviruses and other things in your genome that go up when you age. So that's related to, desilencing that happens stochastically.
It feels like, due to it being correlated with aging and even OSKM, I'm kind of treating this B12 supplementation as getting into a healthier state and then administering OSKM. It's my mental model.
Mm-hmm.
And if you take that stance, and it feels like this is kind of, you have an aged cell, and as it gets older, it struggles to properly do transcription, right? So it's supposed to bind certain areas on your genome and transcribe that one section, but then as you get older, it starts to miss and hit the wrong spots when it's doing transcription. You start transcribing random garbage. You introduce OSKM into that environment where you have an aged cell that is kind of struggling to transcribe properly, and now it's doing a bunch of other transcriptions, like just doing more, right? So doing more work and moving methyl groups around and whatnot. And it's not really surprising then that you start to see more cryptic transcriptions, or it's just missing, right? And so by introducing B12, you're kind of rescuing that generally. You're basically taking the system that was aging and struggling to transcribe properly, getting it into a more healthy state where it has, better amounts of the things that it needs, like B12. And now it's able to properly do what it was trying to do in the first place, which was, do a bunch of transcriptions and moving methylations around and all that stuff. And so that's my working hypothesis. I don't know how to validate that.
Yeah, I mean, certainly when we talk about, cellular reprogramming, you're expressing, or in some cases overexpressing, these factors. So there's an artificiality there. So you're taking, pools of substrates away from other things that are normally happening in these cells.
Yeah.
But if this cryptic transcription stuff is because you need to replenish the pool of methyl groups in the form of S-adenosine methionine for OSK to properly function, just like metabolic pathways for recycling amino acids, if you can rescue this quote unquote cryptic transcription because you supplement with B12, that indicates to me that there's at least for normal operation. Maybe this is you know, by expressing OSKM, you make it seem so obvious because you're really putting a major impact on the accessibility, you know, the availability of methyl groups, you know. But this could be operating under kind of a more finely, you know, tuned system where you're recycling methyl groups, you know, from instead of, you know, from an area that doesn't, you know, isn't critical for silencing, but another area might need to be, you know, importantly silenced. So you have, you know, it could be that these regions are acting sort of as a pool of methyl groups. I don't know. Right. So there's, you know, for a lot of processes in cells, you have intracellular pools of substrates that are synthesized but are kind of waiting in the wings to be used by something. Again, I'm just tossing things out there, right? So if you have actin polymerization, for example, you have more actin. So the concentration of actin that's required for polymerization in inside of a cell is way higher than kind of the, you know, the binding constant of actin, you know, at the end of actin filaments. Meaning that if all that actin was accessible, then your cells would kind of explode because they would make actin filaments everywhere. But most of the actin is actually sequestered in a unavailable form with other proteins, and it's basically doled out as it's required. So, you know, maybe that's the case with areas of the genome, that there's maybe areas that have, again, I'm just speculating here, that, you know, histones that are you know, methylated to a large extent. One of the functions there is to basically form a reservoir of methyl groups for methyltransferases. I don't know. Maybe that's already known. Thoughts, comments? Certainly, the concept of reservoirs of things that are you know, made accessible to enzymatic, you know, functions. That's a known concept in cellular biology. I'm just extrapolating that to methyl groups found in regions of the genome as well. But I might be saying something that's already widely known in the field, or not. I'm not sure. Anybody? Micah, you have a raised hand?
Yeah, we're ready to move on.
Okay.
Yeah. I was wondering, did they do anything to verify that the B12 isn't just affecting their OSKM promotion or expression? Like, I don't know what the promoter on the OSKM in these mice are, but is it possible that the B12 just increases OSKM expression, therefore you get more of it, therefore it does more? They have good data here to suggest that their hypothesis is true. I'm just wondering if they ruled that out somehow.
Good question. I don't know, right? Because you have a transgenic system here. It's in the genome. Genome is affected by methylation status. Is the doxycycline inducible promoter also affected by the methylation status, especially if you're throwing in lots of vitamin B12?
Right, methylation status, or again, if B12 is maybe doing something else that is improving promotion of this, like secondary effect or something. I'm just curious if they ruled this out in any way. Like, they did anything to say we did X to verify that it wasn't.
I don't remember reading that, to be honest. So
I think the way I would test it is just to see, you know, is the amount of OSKM we find in the cells similar with and without B12? If so, it would suggest the B12 is not affecting promotion of OSKM itself.
Yeah, maybe that's a supplemental, but I don't remember reading that in this paper where they basically tested that kind of directly. With and without SAM, with and without B12, look at OSK. Look at then you add doxycycline at the same amount and see whether or not you get higher or lower levels. So I'm not really sure. Okay. So the last thing they do is so they note also that you know, when you have tissue injury, you do have basically you know, some markers indicating some reprogramming is taking place. And last, they basically do sort of an injury model to show that so DSS colitis. So this is a compound that they give orally that sort of basically induces inflammation in the gut. It's, I think, dextran sulfate and something else. I'm not exactly sure how it, induces injury, but it's a model that they use frequently, an artificial model to induce colitis. And this colitis is improved by OSKM or vitamin B12, suggesting that in this model as well, one consequence of increased vitamin B12 and also in conjunction with OSKM is that you have better repair. And the way that they measure repair is, again, some markers of cellular plasticity, such as the SCA1 I mentioned before, which is a marker of intermediate reprogramming. So let's see what they did here. So they note that when they induce colitis, your serum B12 levels go down. So indicating that B12 is being utilized for, and vitamin B12 has also been noted before that it's kind of one of the necessary vitamins to for tissue repair if there's injury. There's others as well, but I think vitamin B12 is one that's noted. Are they looking at cryptic transcription again here as well? So let's see here. So what are they staining here? SCA1. So untreated, mice that were given this bolus, and I believe this is probably, the colon. So SCA1 marker, which is basically indicative of kind of cellular plasticity is happening, goes up if you add B12, goes up if you add OSKM. And this is an inhibitor again, so it's of kind of the availability of methyl groups. So that goes back down again. Let's see, does it go up with, so OSKM, so would have thought they had B12 and OSKM, right? So I don't really see that there. So do they do B12, does it improve if you add B12 and OSKM, or is it just, do they look at B12 separately from OSKM? Let's see, histologic score.
Separate only. It looks separate only.
Yeah, it looks separate only. So I don't know why they didn't combine it with OSKM.
They're basically just saying B12 good.
Yeah, B12 good. So take your pick, right? So you can improve. So let's see. So you can certainly improve, at least reprogramming by measuring the SCA1 marker by adding either B12 or OSKM. But you would think you would get based on what we saw earlier, a better result if you have both. Let's see, did they mention it, or is it? There's no figure here for that. I thought they had both as well. Doesn't look like it. Let's see, where's the text for that? DSS-induced colitis. Mice were treated with 2.5% DSS. Colitis grade. There's a colitis grade. Let's see, 5 at day 5 DSS removed. Oh wait, no, I'm just reading the materials and methods.
And that actually did mention it, I think, what you were reading just now.
Oh, inside the materials and methods?
Yeah, it started to in the second half of that paragraph under DSS-induced colitis. Mice in the B12 experimental group also received supplemental vitamin B12. This was right after the line that said doxycycline was put in the drinking water. So the phrasing is a little odd, but you could interpret it either way.
Yeah, it looks like it overlaps. So OSKM.
So I think that's DSS plus B12 and OSKM, and DSS plus B12 plus OSKM plus MAT2AI.
Yeah. So is this DSS plus B12 plus OSKM?
I mean, I really feel like they should have put that in the label, but
I mean, it looks like it from figure A, right? I mean, there's...
the DSS plus B12 is just DSS plus B12. I think maybe the DSS plus OSKM is also with B12. They left
Yeah,
it out,
'cause,
but then they
I mean,
included again.
yeah, it looks like B12 is there regardless of whether it's OSKM or not, right? It's I mean, unless you specifically say that you don't have it right here. Is there, so is this B12 without OSKM? Let's see. Source figure legend here. D. SCA1 immunohistochemistry on colonic sections on day 9 after DSS with the indicated treatments. Untreated control, DSS plus vitamin B12, DSS plus OSKM. All right, so I'm going to assume, I'm going to ignore that figure up here. See, colitis was induced by DSS followed by a 48-hour pulse of OSKM or continuous administration of vitamin. Oh, okay. All right, so these are separate. These are not overlapping. Never mind.
So that's interesting.
Yeah. All right. So just so this is either OSKM or vitamin B12.
Moral of the story: take your B12.
Yeah. Okay. So anyway, I'm gonna stop sharing here. So B12 looks like it's and probably more directly, SAM is definitely necessary. Although they didn't test SAM here for improved kind of repair from colitis, I would think. And I don't know how bioavailable SAM is. I know there's supplements for it, but I don't know. Doesn't mean that you actually, how much of it actually gets through your blood-brain barrier, not blood-brain barrier, through your gut barrier into your bloodstream. People sell supplements all the time, but
A quick search suggests that it does have some amount of bioavailability, like a useful amount. It's not one of those things that just doesn't make it anywhere at all. It does get into your bloodstream. You can detect it, and you can take a gram of it, and it'll show up in your system.
Right. So
I don't know how that compares against B12. Maybe B12 is easier.
Yeah. So one thing to look at least from an in vivo perspective, right? Is it better to take, even though SAM is more downstream, is it better to take B12 because you get 1,000,000 times more B12 into your bloodstream versus taking in S-adenosine methionine and you get 1,000,000 times less in your bloodstream, right? And that translates to B12 actually generating intracellularly 10 times more SAM than SAM getting in through the gut. I don't know. Again, these are these types of, I feel these types of papers and these types of experiments when it comes to accessibility of micronutrients and metabolic factors. There's a lot of these papers where people have been studying basically transport across the gut into the bloodstream. I think given the many decades people have been taking supplements, there's been a whole slew of papers that have been looking at measuring blood levels. So I'm sure those papers are out there. I'd be shocked if they weren't. Where you can basically look at the bioavailability of a variety of these things and what would be better to take, right? If you're looking for improved reprogramming or improved tissue repair as a consequence.
Do we have the ability to detect SAM levels within a cell or within a population of cells?
I would think so.
If so, then it seems pretty simple. I mean, you just have a human supplement some B12 and have another human supplement some SAM. Do that a bunch of times and test to see which of their cells has more SAM in them at the end.
Are you talking about like through biopsies, like looking at actually cells?
Yeah, biopsy, or pick some cell type that's easy to extract. Blood, maybe.
Yeah. Line blood's always the easiest. Yeah. Again, this is something we can jump on PubMed and basically do a bunch of keyword searches and see everything that's been noted regarding circulation and bioavailability of each of these B12, SAM, and anything else that's related to 1-carbon metabolism, and specifically for the generation of methyl groups for enzymes that require them. Okay, well, take-home messages is eat your vitamins. Make sure you have plenty of them, particularly B12 if you want to repair your tissues, and that we can through a very simple addition here, you can dramatically increase the ability of cellular reprogramming factors to function by basically I mean, in one sense it's not a surprise, right? You're boosting levels of a substrate that they require, acetinosyl methionine, right? But in another direction, it's I guess heartening to note how easy it is to do that with a simple supplement that's readily available. So that's something to keep in mind, and something that I'll keep in mind when I read more reprogramming papers to see what other tricks can be done to to boost their efficiency. Because I don't recall reading in other papers where they used OSK factors to reprogram, where they explicitly stated that they're also supplementing with SAM or B12 or other things to enhance. Right? You look at doxycycline induction, OSKM, but it's left at that, and then the mice are fed with their mouse chow, whatever it is, and you assume that all your vitamins and minerals are there, but maybe they need to drastically improve B12 and while they do these experiments, or maybe not, right, because we're talking about partial reprogramming, so you don't want to go too far with reprogramming, so maybe
the reduction in cryptic transcription makes me think that it's not necessarily that it's making the reprogramming happen faster, but it might be this making it happen more efficiently and accurately.
Could be, just because if there is a pool, again, I'm going to look into this to see if the cryptic transcription is a consequence of a pool of methyl groups being shunted from an area that's kind of acting as a reservoir to OSKM factors, and yeah, I don't know. Gonna do some searching to see if that's the case.
So a just quick follow up. I did 30 seconds more searching on SAM versus B12, and it does look like SAM is not as available as B12, which is when doctors want you to get more SAM in your cells, they tell you to take B12, as apparently it is a better route.
Makes sense. Yeah, makes sense. But if you're talking about reprogramming in a tissue culture dish, maybe SAM is the way to go if it's available to be transported across the membranes of individual cells more readily. Yeah, especially, well, certainly given the case where they did, maybe that's why they didn't do the SAM in vivo with mice, but they used the SAM in the murine embryonic fibroblasts in culture. Okay, well, that's all I've got to say on this paper. I think this was a pretty good contribution to the field of cellular reprogramming. Sometimes, something like as simple as that could, really help spur things along, especially since it seems to have a really significant effect on improving the ability of OSKM to basically reprogram parts of the genome that we want reprogrammed. So with that, Steve, when's our next journal club? We're getting into holiday season here.
Great question. My wife asked me that a few minutes ago, actually.
Early?
Well, it's going to be a bit earlier, isn't it? Because we are going to be doing a traditional Christmas close down. The last day is Friday the 22nd, and we are back on the 2nd for the news and pretty much everybody at Lifespan, because even we need a break, right? I mean, we can't be working every waking hour. So, I think it's probably maybe the 15th, so not very long away at all. couple of weeks.
All right. So if there's any suggestions for papers you all want to do, send them my way, send them Steve's way. Otherwise, we'll pick something that we like, as per usual, and we'll talk about it. Something appropriate to the subject. And I'm actually running out of time. I got to run out the door in a few minutes. Usually, I love to ramble on with everybody here and talk about applications. But in the meantime, make sure you have adequate supplies of vitamin B12 in your diet, especially if you're going to be hitting the eggnog extra hard. And and Ollie says I'd suggest the paper I sent earlier. So we will take a look at that. So let's see. I got the link right there. All right.
Yeah, so it'll be definitely, I think, going to be a couple of weeks' time for that, and then we can look forward to that, and
Okay.
and then we can all have a nice holiday season, as you guys call it, holidays.
Yep.
We just call it Christmas. That's it, really. I don't know about you guys, but I'm looking forward to it. So we've been working hard this year, and yeah, I think we've earned, all of us. I think we've earned a bit of time off. So yeah, have your turkey or your tofu or whatever it is you fancy. So
Tofurkey. Okay, everybody, take care, and Ali, I'll take a look at that paper. All right. See you, everybody.
Okay,
Yep.
thank you. Bye.
Take care, everyone.
Thanks.